So this protein energy, so this is the, now we are discussing about the polymerase chain reactions. So polymerase chain reactions, we are using thermus aqueous Taq polymerase enzyme. So DNA, what are the targets? Then also targets, denaturation, primers, annealing, primers like cycles, requirements. What are the requirements? DNA detection factor 5, then PCR results also factor 5 conclusions like this. DNA is nucleic acid that composition of two complementary nucleotides building blocks chains. The nucleotides are made up of phosphate group, 5 carbon sugars and nitrogen base. DNA sugar, deoxyribose nucleic acid, RNA sugars, ribonucleic acid. So DNA has 4 nitrogen bases, 2 are purines, 2 are ringed bases, and adenine, guanine, 2 are pyrimidines, like 1 ringed like cytosine thymidine. The 4 bases are linked in a repeated pattern by the hydrogen bonding between the nitrogen bases. The linking of the 2 complementary strands is called as hybridization. A purine always links with a pyrimidine basis, maintain the structure of DNA. Adenine binds to the thymidine with the 2 hydrogen bonds between them. Guanine binds with the cytosine. There are 3 bonds between them. So examples of pattern like primary strand and complementary strand. The first one is primary strand, second one is complementary strand. So adenine, thymine, guanine, cytosine. These are the bases of DNA. Base pairs. Then also PCR is a technique that takes specific sequence of DNA, small amounts, and amplifies it to be used for further testing. The targets in PCRs are sequence DNA on each and end of the region interest, which can be a complement gene or small sequence. PCR target, like the number of bases in the target can vary. So this is how the target DNA sequence. So denaturing is the first step of which is denaturation means that is 95 degrees heating. So ultimately that temperature will be increases, that the melting is also here occurred. The primers range is 15 to 30 nucleotides are single stranded and are used for the complementary building blocks for the target sequence. So a primer for each target sequence on the end of the DNA is needed. That allows both strands to be copied simultaneously in the both directions. Here is simultaneously both directions. The primers are added in the excessive, so they will bind to the target DNA instead of the 2 stranded binding back to each other. Annealing is the process of allowing 2 sequences of DNA to form hydrogen bonds. The annealing of the target sequence and primer is done by the cooling the DNA to 55 degrees centigrade. Taq strands, the thermus aqueous, which is microbe found in the 170 degrees Fahrenheit liquor hot spring in Yellowstone National Forests. Taq produces an enzyme called as DNA polymerase that amplifies the DNA, the primers polymerase chain reactions in the presence of magnesium. So how it is shown diagram of PCR cycle. So simultaneously both the semi-conservative model here, in that PCR showing denaturation 94 to 95 degrees, primer annealing and 55 to 65 degrees, and the extension like a 72 degrees temperature. Number of cycles like 25 to 40 cycles. So this is the requirements: magnesium buffer, DNTPs, primers, DNA polymerase targets, and DNS. So one of the Neisseria, gonorrhea, chlamydia, these are the factors we can see in these things. Some of the Neisseria. So how these applications like swab, HIV, all those things we can discover. Then coming to the how this showing of structure, like this is the structure. Then also extractions of factors. How diagram shows this extraction, how we can insert with the structures. These structures will be very important here. So how the picture is showing means, so we can inject with the simple with the thermocyclitis, that is wells. So the structure is important here. These are the reference we can discuss to the PCR. These things. Thank you very much.